Hi all, welcome to Physio Exam Buddy. This video is on the concentration of urine. For human body to produce concentrated urine, there are two basic requirements. First one is the presence of antidiuretic hormone and the second one is a hyperosmolar medullary interstitium. In the presence of ADH or antidiuretic hormone, the water permeability is increased in the DCT as well as in the collecting duct, which will favors water reabsorption in these areas. So water will be reabsorbed back into the interstitium. So solute rich urine will be excreted, that is concentrated urine. Second one is the hyperosmolar medullary interstitium. This hyperosmolar medullary interstitium, it provides an osmotic gradient which aids the water reabsorption in the presence of high level of ADH. So we will see how this hyperosmotic medullary interstitium is produced in the kidney. You all know that the osmolarity of the interstitial fluid in all parts of the human body, it is about 300 milliosm per liter. And you know the two parts in the cross section of the kidney. The outer one is the cortex and the inner one is the renal medulla. So the osmolarity of the interstitial fluid in the renal medulla increases from 300 to 1200 from this corticomedullary junction to the tip of the papilla. And this progressive increase in the osmolarity, it is with the help of the counter current mechanism. What is this counter current mechanism? It basically occurs in U-shaped tubes. That is where the inflow runs at the parallel to as well as counter to and in close proximity to outflow for some distance. You know that the loop of Henle as well as the vasa recta, they are in the shape of a U. So the inflow of the fluid occurs through this limb and it occurs parallel to the outflow as well as counter to the outflow. That is, here inflow flows in this direction whereas outflow flows in the opposite direction. And these two limbs should be in close proximity. That is, the inflow should be in close proximity to the outflow at least for some distance. And in case of renal medulla, this occurs both in the loop of Henle as well as in the vasa recta. So, this increasing osmolarity in the renal medulla, it is established with the help of the loop of Henle. So, this loop of Henle is commonly referred to as the counter current multiplier. Whereas the osmolarity established by this loop of Henle, it is maintained by the vasa recta, which is running parallel to the loop of Henle. So, this vasa recta is commonly referred to as the counter current exchanger. Now we will see the function of the counter current multiplier, which is our loop of Henle. There are four main things happening in the loop of Henle. That is, this is the descending limb and this is the thin ascending limb and this is the thick ascending limb. The thin descending limb, it is highly permeable to water and water moves out mainly by passive diffusion here in the thin descending limb. Whereas in the thin portion of the ascending limb, there occurs passive transport of the sodium chloride. Whereas in the thick part of the ascending limb, there occurs active transport of the sodium and chloride out to the medullary interstitium. Along with that, there occurs an inflow of the tubular fluid from the proximal tubule to the outflow into the distal tubule. So these are the four main things that events that is happening in the loop of Henle. So how it will multiply the osmolarity in the interstitium by counter current multiplier mechanism, we will see. So these are the hypothetical steps in the formation of the hyperosmolar medullary interstitium, and this picture you are supposed to draw along with the counter current mechanism. So we will see in each loops what is happening in detail. So this is the first loop. So you can see that the osmolarity, it is same in the descending limb, ascending limb as well as in the renal medullary interstitium. So we are first assuming, I have already told that these are hypothetical steps. So you are first assuming that the osmolarity of the loop of Henle as well as in the medullary interstitium, it is 300 milliosm per liter. Now I have already told that the thick part of the ascending limb of loop of Henle, it actively transport or pumped out the solutes, particularly sodium and chloride, into the interstitium. So this active pumping, it is represented by a circle as well as the arrow indicating the direction of the flow of the solute. So the thick ascending limb of loop of Henle, it actively pumps out around 100 milliosum of the solutes, and always the gradient produced between the thick ascending limb of loop of Henle as well as the medullary interstitium, it is 200 milliosum. So the thick ascending limb of loop of Henle pumps out 100 milliosum of the solutes. So what happens to the gradient? 100 from the 300, 100 goes here. So here the 300 increased to 400, and here the 300 is reduced to 100 because the 100 is pumped out of the thick ascending limb into the medullary interstitium. So this pumping out of the sodium chloride occurs till a gradient of 200 milliosum established between the tubular fluid as well as the medullary interstitium. Now what happens here? You got an increase in the osmolarity of 400 because of the pumping of the sodium and chloride. So in order to maintain the equilibrium, the water will move from the thin descending limb of loop of Henle till the contents equilibrate with the interstitium. So here the movement of the water is by passive transport. That is why it is by shown as dotted arrows showing the direction of the movement of the water. By this time, there occurs additional flow of the isotonic fluid, that is 300 milliosum per liter, from the PCT into the loop of Henle. So the hyperosmotic fluid in this descending limb, it will flow into the ascending limb, and the hypotonic fluid flows into the DCT. Again, the steps will be repeated. That is, the hypertonic fluid reaches when the hypertonic fluid reaches the ascending limb, more and more ions, particularly sodium and chloride, will be pumped out by active transport until again a gradient of 200 milliosum is established between the tubular fluid as well as the medullary interstitium. You can see here the difference between the osmolarity. Here is 200, again 500 minus 300, it is again 200. So the hypertonic fluid reaches ascending limb of loop of Henle, and more ions are pumped out until a 200 milliosum gradient is established. And you can see here that the interstitious osmolarity rises from 300, then 350, then into 500. Now, in order to maintain the equilibrium, what happens? Fluid in the descending limb, it should move out into the medullary interstitium, equilibrating the contents with the equilibrating the contents of the tubular fluid with the medullary interstitium. So here the passive transport of water is again shown. So these steps will be repeated again and again until the small gradient at each horizontal levels become multiplied in vertical direction. So because of this counter current flow, as well as since all these events from 1 to 6 are repeating, this gradient is established from 300 to 1200 milliosum per liter. So this is how the hyperosmolar medullary interstitium is created by the counter current multiplier, which is the loop of Henle. So this result in a large medullary gradient from the top of the outer medulla to the depth of the inner medulla. Now what happens when the length of the loop of Henle or the loop increases? The osmolarity which is reached at the tip of the loop of Henle will be more. So greater the length of the loop, greater will be the osmolarity reached at the tip of the loop of Henle. So in case of cortical nephron, the osmolarity attained at the tip, it is around 600 milliosum per liter. And you all know that the juxtamedullary nephrons, it has a very long loop of Henle, and it will enter deep into the medulla. So in that case of juxtamedullary nephron, the osmolarity at the tip of the loop of Henle can even reach up to 1200 milliosum per liter. Next factor which contributes to the countercurrent multiplier mechanism is that in case of juxtamedullary nephrons, that is, this mechanism is operating only in the juxtamedullary nephrons. Here, the thin thin ascending limb of loop of Henle, it is actually impermeable to water. But you have already seen that it is passively permeable to sodium and chloride. So the sodium and chloride will move down the concentration gradient from the thin ascending limb of loop of Henle to the interstitium, which will contribute to the additional passive counter current multiplication. But please remember, this happens only in juxtamedullary nephrons, not in cortical nephrons. Next role is by the distal tubule as well as the collecting duct in the concentration of the urine. We all know that the fluid which is reaching the distal convoluted tubule, the osmolarity ranges from 100 to 140 milliosm per liter. And this DCT actually functions like our ascending limb. So early DCT further dilutes by actively transporting the sodium and chloride out of the tubule into the interstitium, and this portion is again relatively impermeable to the water. Now, when the fluid reaches the cortical collecting duct, there the water reabsorption, it is mainly dependent on the ADH. In the absence of ADH, it is highly impermeable to water. So, there occurs continuous reabsorption of the solutes. So, in the absence of ADH, it results in the formation of a dilute urine. But what happens in the presence of ADH? It will be highly permeable to water. So, more and more water will be reabsorbed from the tubular fluid into the medullary interstitium, producing a concentrated urine. When high levels of ADH are present, the collecting ducts become more permeable to water, and fluid at the end of the collecting duct has the same osmolarity as the interstitial fluid of the renal medulla, that is around 1200 milliosm per liter. Next is the role of urea recirculation in the concentration of urine. Around 40 to 50% of the medullary osmolarity, it is contributed by the recirculation of urea. In the proximal convoluted tubule itself, 40 to 50% of the filtered urea from the glomerular filtration is reabsorbed back. Now, when the fluid reaches the thin descending limb, urea is actually secreted into the tubules from the interstitium. So, the urea concentration in the tubular fluid will increase. Now, when the fluid reaches the thin ascending limb, again urea is secreted, which will further increase the concentration of the urea in the tubular fluid. But when the fluid reaches the thick ascending limb, DCT, as well as in the cortical duct, which is actually impermeable to urea. And when in medullary cortical collecting duct, it is the facilitated diffusion of the urea that is happening. So you can see that basically it is actually recirculating within the renal medullary interstitium. So, in the cortical collecting duct, in the presence of ADH, water will be reabsorbed and the urea concentration further increases. And in the medullary collecting duct, the specific urea transporters, that is UTA, will facilitate the reabsorption of urea into the medullary interstitium. So, this ADH also stimulates the urea transporter A1 as well as A3 transporters and further increases the reabsorption of urea. So some urea from the interstitium diffuse into the thin loop of Henle and pass upward and back to the medullary collecting duct. So this contributes to the 40% of the medullary interstitial fluid osmolarity. So this type of urea recirculation, it happens several times before the excretion. Now we will see the factors maintaining the gradient. So till now we have studied how the hyperosmolar medullary interstitium is created. Now we are going to study how the osmotic gradient is maintained. Two factors which helps to maintain the osmotic gradient. First one is low medullary blood flow. That is, the blood flow in the renal medulla, it is less than 5% of the total renal blood flow. Whereas the other factor which maintains osmotic gradient, that is one of the most important thing, that is a vasa recta, which acts as a counter current exchanger by minimizing the solution washout from the interstitium. We will see the function of the counter current exchanger. So this vasa recta acts as the counter current exchanger which lies very close to the loop of Henle. So blood is the one which is entering the vasa recta. So as blood in the vasa recta descends downwards, there occurs entry of the solutes, particularly sodium chloride as well as urea from the medullary interstitium. Whereas what happens to the water? Water will be leaving out of the vasa recta. And as the blood in the vasa recta ascends up in the cortex, exact opposite happens. That is, solute will diffuse into the interstitium from the tubular fluid and water will enter into the vasa recta. So it becomes less concentrated. So you can see here that the solute is actually recirculating within the medulla and hypertonicity is maintained. This is the next diagram you have to write along with the concentration of urine as well as the counter current mechanism. Next one is the medullary blood flow, which helps to maintain the osmolarity of the medullary interstitium. What happens when the medullary blood flow is increased? It will cause the washout of our solutes and decreases the urine concentrating ability. But these are the factors which increases the medullary blood flow. That is vasodilators as well as increase in the arterial blood pressure. So, in short, about the concentration of the urine, you have to explain the counter current system. That is both the counter current multiplier as well as the exchanger. Next is in case of juxtamedullary nephron, you have to explain the role of ascending limb, which is contributing to the additional counter current multiplication. Then the third one is there is a role of the DCT as well as the collecting duct in the concentration of urine. Then the role of urea recirculation and the role of the medullary blood flow. So this is how you have to write the answer of the concentration of the urine. And if the question is only on the counter current system, just explain the counter current, making the multiplier as well as counter current exchanger. Draw the two diagrams, that is the hypothetical steps diagram as well as the diagram of the vasa recta where the solutes as well as water is recirculated between the ascending as well as the descending limb. Thank you.